Welcome. I'm Sean McKenzie. I'm Director of CLF, and I want to welcome you to the 21st Annual Dodge Lecture. Welcome. Today's lecture is very special for many reasons, and one of those reasons is that this marks the first in-house event that CLF has hosted in quite some time. So I want to give a round of applause to our staff for making it happen. And we normally thank people at the end, but I do want to mention a few people who have really made a difference to pull this off today. In particular, Veronica Green. Veronica, I'm not sure if you're where you are, but thank you. Jossie Rayner, of course, always there, making it happen. Natalie Woodwright, who I believe is online and always has the magic touch to keep our communication going. Thank you, Natalie. Ruchiro Drain, who is here from Northern California. Thank you, Ruchi. Also, of course, Christine Grillo, who's busy documenting this event even as I speak. Thank you, one last person I do want to point out is our one and only Mike Milley, who's multi-talented, including being a great photographer. I hope someone gets a photo of him doing his work today. We can do that. But last but not least, I do want to give particular mention to someone who I know will be angry that I do this, but we have a new program officer with CLF who organized this whole event. Her name is Tiffany DeFreitas, and I want to acknowledge her. Thank you, Tiffany. Although, as Tiffany pointed out before, the event's not over, so we'll see how it goes. I do also very briefly want to acknowledge some of our distinguished guests in the audience, including our very own departmental chair, Marcia Wills-Karp is here. Well, Lance will get his own special introduction soon, but I want to recognize Lance's family. Cindy Liu, a remarkable researcher in her own right, and Lance's life partner and partner in crime, I believe, as well. As well as Cindy's parents, Dr. and Mrs. Liu, are here today, so welcome them. Finally, another distinguished guest we have today is our very own founding director and professor emeritus, Bob Lawrence. So, and I'm going to actually hand off to Bob because, as many of you know, we lost a good friend at CLF. Ed Dodge passed away last fall, and two members of his family are here, Randall and Colleen Dodge. And I think to get things started, I will hand off to Bob Lawrence, who will share a little bit of his perspective and memories of a good friend, Ed Dodge, and a good friend to CLF. Bob Lawrence.
Thank you very much, Sean, and it is wonderful to be back post-pandemic, and I hope there will be many more CLF sessions for entice me down from mid-coast Maine. But today it is really a great pleasure to acknowledge Ed Dodge. When Ed passed away last September, I was contacted by one of our loyal staff members in the CLF about a memorial note remembering and thanking Ed for his support. And I drafted comments and a quote, and I said in the quote that perhaps part of the reason we bonded so early and so well is that we were both PKs. And the email came firing back, "What's a PK?" So a preacher's kid. And Ed was the son of a Methodist bishop who served in Angola, Mozambique, and later Zimbabwe as well. And Randall remembers his grandfather well, in fact, visited him in East Africa before his grandfather passed. Ed started out with a intention of doing medical missionary work influenced by his father. and he went to medical school at Indiana University. Then he actually started a surgical internship in California and realized early on, as many surgical residents do, that maybe this was not the road for him. So he left after his internship, and he then came later to this school and got his MPH in 1967. Then he realized part of his dream of being a medical missionary, but as a public health person teaching at the Haile Selassie University in Gondar, Ethiopia. And after a few years there, returned to public health work in Florida, and then as a practitioner, primary care for most of his career. But the reason that Ed is so important to us at the CLF is that after we'd been in operation for about a year and a half, enough seemed to have happened in 1997 or so 98, that a short note about the work of the CLF was posted in the Hopkins magazine. Ed, as a alumnus, received it, liked what he read, and unsolicited sent a very generous gift and announced that if Dodge Family Foundation was going to support the CLF a certain amount in perpetuity. Well, that bowled us over. It was the first sort of external affirmation that maybe we're on the right track. But Ed's gift was really unsolicited and with no strings attached. And it was a huge morale booster. And we decided to set aside a good portion of the annual gift to endow this lectureship. And we have over the years enjoyed some remarkable people. I won't intrude on Keith's introduction of today's speaker, but this has been a really distinguished list of people we've been able to bring to the school. And for that, we'll be ever grateful. Ed and I enjoyed a close friendship, frequent communication over the years, and it was really with great sadness that I learned of his death last September. But I'm so pleased that Randall and Colleen are here representing the family, and we hope in the future that they'll be joined by Ed's other surviving children, another son and a daughter. So thank you very much for being here. Thanks for coming from Indianapolis, and I'll turn it over to Keith.
Thank you so much, Bob. Hi, everybody. My name's Keith Nachman, and I'm the Associate Director of the CLF and the Robert S. Lawrence Professor of Environmental Health and Engineering. Yes, that Robert S. Lawrence. It's actually a special moment to be here. First time we've been together in person since that professorship naming, so thanks. It's my honor and pleasure to introduce our 21st annual Edward and Nancy Dodge Lecturer, Dr. Lance Price. By way of background, Lance earned his bachelor's and master's degrees from the Northern Arizona University in microbiology and biology, respectively. He completed his doctoral training here at the Bloomberg School of Public Health in the Department of Environmental Health Sciences, which we now know as Environmental Health and Engineering, under the mentorship of Dr. Ellen Silvergeld. After finishing his PhD, Lance returned to Arizona to establish and lead the Center for Food Microbiology and Environmental Health and the Center for Microbiomics and Human Health at the Translational Genomics Research Institute in Flagstaff. In 2012, Lance felt the East Coast beckon him back and accepted a position at the George Washington University Milken Institute School of Public Health as a professor of environmental and occupational health, where he founded the Antibiotic Resistance Action Center, which he still runs today. Lance is having a remarkable career focused on identifying and addressing the drivers and impacts of the antimicrobial resistance crisis. While his academic record is impressive, with nearly 150 peer-reviewed publications, I rounded up a little bit, and a robust record of funding from the NIH, NSF, and numerous philanthropic sources, equally or possibly more important is Lance's track record of effective contributions in public health policy and communicating. Over the last 15 plus years, Lance has been a leading voice for public health on the matter of antimicrobial resistance, particularly in the domain of the misuse of antibiotics in the context of food animal production. He's provided invited expert testimony for the US Congress and state legislators, advised US, Icelandic, Danish, and other government agencies, served on high-level international panels at the World Health Organization, and helped develop industry procurement policies for responsible antibiotic use. He also served as an advisor to the landmark Pew Commission on Industrial Farm Animal Production, which many of us are familiar with, which played a really important role in changing the conversation on agricultural antimicrobial use. Lance has also worked closely with numerous high-profile media outlets to raise awareness and spur action on the issue of antimicrobial resistance. Building on his many, many appearances in the New York Times, the Washington Post, CNN, PBS NewsHour, 60 Minutes, I got more, I'll stop though, and many others, he recently appeared in Netflix series Poisoned, The Dirty Truth About Your Food. Personally, I can share that I'm happy to live in a world where I can tune into Lance Price singing the public health gospel on demand in my streaming services. Before I turn things over to Lance, I wanted to take a moment to highlight today's significance from a slightly different angle. We're proud to be hosting the 21st Distinguished Speaker of the Dodge Lecture, and Dr. Price here is a really terrific addition to a quite remarkable series of leading thinkers in the domain of food systems dedicated to addressing the public health implications of ecosystem change resulting from personal and policy choices. But this is also a special moment for CLF because Lance's connection to us and at the center goes back more than 20 years when he was one of the first awardees of the CLF Learner Fellowship. Lance, on behalf of everyone at CLF, please know that we've enjoyed following your career trajectory, have greatly valued your important contributions, and we're really proud to call you one of our own. So without any further ado, I'd love to invite Dr. Price to the podium. Sound pretty good on paper. I don't know. We'll see how it goes. Thank you so much. Thank you so much for having me today. This is a real honor and a bit of a surprise. If you look at the past speakers like Vandana Shiva and Marianne Nessel and Wendell Berry, I think there's been a mistake, but we're just gonna, it's too late now. Let's keep going with it. But I will say that one of the most inspiring lectures that I saw as a student here was Fred Kirschman. So it was the 2006 Dodge lecture. And it was both the content of what Fred spoke about and the way he spoke so clearly that inspired me and has really influenced the way I've spoken about science ever since. And so I thank the Dodge family so much for this contribution and all the training and education that you're giving to people. So with that, I have some disclaimers. We're a little bit stuck here. But so, yeah, here we go. So the disclaimers are that I spent a lot of time playing with these kinds of toys as a kid. And so this really did influence my view of what farming looked like. We also have a cattle ranch in Central Texas, but that's a cattle calf operation, pretty natural. And then the other disclaimer is that I spent a lot of time in my career trying to influence these people. And when you talk to these people, you got to keep it simple. So these are, in my talk, these are human beings. If you see something like that, that's a bacterium. If it has a lightning bolt, it's antibiotic resistant. And one of the first things that I always have to tell the policymakers is that bacteria become resistant, not people. Okay, so the other disclaimer is that I'm a harm reductionist, right? So I believe in needle exchange programs for heroin addicts. I believe in free condoms for commercial sex workers. And I believe that, food animal production is something that we have to deal with, and it has a long list of harms. I don't think I can single-handedly, take this thing apart, but, I can work on one of these harms and I can make a difference. And that's what I chose to do 20 years ago. And this is an important topic to work on, or an important subject, or harm, because as the World Health Organization said just a few years ago, they said, look, the world's headed for a post-antibiotic era in medicine, a time when we can no longer rely on antibiotics to cure even simple infections.
And this is going to change medicine. And so they're warning of this time in the future. Just 2 years later, the CDC comes out and they said, hey, we've got to quit talking about a coming, post-antibiotic era. It's already here. We are living in a time when when miracle drugs no longer perform miracles and families are being ripped apart by microscopic enemies. And the time for action is now. Well, CLF had vision, 20 years ago that the time for action was back then. But unfortunately, today, or 2019, more than 1 million people are dying of drug-resistant infections around the world. And what we know is that improving antimicrobial stewardship, that is how we use antibiotics, is one of the keys to changing this trajectory that we're on. And so, we know that 70% of antibiotics roughly are used in food animal production, and maybe only 30% are used in human medicine. But I don't think that proportion translates to 70% of human infections that are drug resistant are coming from food animal production. It's worth trying to quantify, though, what is the contribution so we can optimize our interventions. Because if this doesn't matter, if the animal production doesn't matter, then let's just focus our money on the humans. And so this was me 20 years ago in my favorite lavender shirt. A couple friends in the back probably remember that shirt. Wore it a lot. So I was one of the first 2 CLF fellows. And when I started with the center, I started with really simple questions. Kind of a simple dude. So first question was, does meat from animals raised without antibiotics have fewer antibiotic-resistant bacteria than conventional products? Well, the answer was yes, right? So we tested it. It turns out that there was a lot less Cipro-resistant Campylobacter. Cipro is a really important antibiotic in human medicine, and they were using a drug called enrofloxacin that's exactly like ciprofloxacin, but has 1 extra hydrogen. The microbes don't recognize the difference. And so our use of enrofloxacin was fueling drug-resistant infections in people, but also, the vehicle was food. Now, this kind of meat comparison, looking at this, has been carried forward by other generations of CLF fellows, including Megan and Gabriel. And I've worked with them on this, but they've taken it to another level where they're FOIAing, they're requesting government data on the National Antimicrobial Resistance Monitoring System, and they're really analyzing these data deeply, and it's holding up. I mean, you use antibiotics, you get drug-resistant bacteria on the food. So the next question we looked at was looking at the people who catch chickens for a living. These are chicken catchers, clever name. And I figured if anybody's going to pick up drug-resistant bacteria, it's got to be these guys. So we just asked that question, are they at excess risk? And we did the study. We went out to the Eastern Shore with Keith Nachman, who sang War Pigs in a bar on the Eastern Shore. It was amazing. But we recruited a lot of poultry workers into the study, and we asked them for stool samples. And we looked at the rate of carrying drug-resistant bacteria in this population versus their peers. And it turns out they had 32 times the risk of carrying gentamicin-resistant E. coli. Now, when you get numbers like that, you have to think, well, I made a mistake. So you go back to the numbers checked out. And that's when we learned about this. This is an in ovo injection device. So they were injecting pallets, literally billions of eggs with gentamicin on their way to the hatchery to prevent infections. And so it made sense. So I learned a lot through this study. I also learned a lot about chicken catchers. These guys have massive hands because they can catch, 4 birds in each hand, and they have big shoulders from throwing those birds into these cages, and they can poop on command. These guys would just come in, we'd give them a gift certificate, and they'd say, you need a stool sample right now? Where's the bathroom? It was amazing. So we got a lot of great samples from these, and these guys, I really appreciate it. The other thing that we know is that 9 billion with a B, chickens produce a lot of chicken waste. Dr. Nachman's done a lot of work, Nachman has done a lot of work looking at arsenic and chicken waste. But Jay Graham asked this question. So Jay was another CLF fellow, asked this question, do the drug-resistant bacteria persist in the animal waste that's being used as fertilizer for crops? And so he did the research. I collaborated with him on this. And sure enough, the drug-resistant bacteria were lasting for months in the poultry waste, in the compost, way past the time when they said it was safe to apply to even crops where the edible portion of the crop was touching the soil. So I felt like we were making really good progress, but for some reason, the infectious disease communities still were dismissing this issue. And I think part of the problem was that most of the research that had been done up to that time had to do with Salmonella and Campylobacter, some of the classic foodborne pathogens. And so even some of our research was on this. Yeah, they make stuffies with life-threatening diarrheogenic pathogens. It's pretty awesome. I was looking through my slides this morning. My daughter goes, "Oh, yeah." Anyway, so we were framing too much on this. And when we talked to our really good-looking clip art doctors about infectious diseases, they want to know about E. coli causing urinary tract infections, not diarrhea. They wanted to know about methicillin-resistant Staph aureus. And with good reason, these are the top killers in the world. Right? So top two pathogens killing people around the world are E. coli and Staph. They are also the top two pathogens directly associated with fatal antimicrobial resistant infections. So they were right. I had to study this. And the crazy thing was that when we cultured meat, we were finding lots of Staph, lots of E. coli. In fact, more than we were finding of Salmonella and Campylobacter. And so I had to ask, why were people ignoring all this contamination? And it turned out that it seemed like the industry was really controlling the discussion, right? So the industry was saying, hey, those are different E. coli, those are different Staph, don't worry about that that's in the food. But I started looking for the data, and the data were not there to support those arguments. And so when I left here, I went to a place called the Translational Genomics Research Institute, a nonprofit research institute based out of Arizona. And this is a really cool looking building. We were actually in a strip mall because we were in the Flagstaff location, but it doesn't make as good a slide. And we had some really early successes using genomics to study the evolution or the emergence of livestock-associated MRSA, methicillin-resistant Staph aureus. So while we were coming through this program, they were just discovering this livestock-associated MRSA, and this is ST398. That means sequence type 398. Never been seen before, but in 2004, they found an infant with a MRSA infection. And they traced that infection back to her family, who were pig farmers. And so we learned that it was, farmers were at excess risk, farm workers, veterinarians that worked with large animals, household contacts of these people. There was a dose-response relationship. So the people with the most exposure had the most likelihood of picking these infections up. And in some European communities, they were 1/3 of MRSA infections were these livestock-associated strains. So we applied this emerging technology of whole genome sequencing to understand where these things were coming from. Where had this come from? And so we sequenced a bunch of these genomes. It was, I think 100, which was a big deal at the time. It was a global collection of these isolates. And we showed that the methicillin resistance had really emerged within the swine population. So this was not MRSA introduced from people into pigs and then spilling over. It emerged in those swine herds. And we published this paper. And this paper really caught fire, and it's had almost 850, I'll round up, 850 citations. It was covered in Discover magazine as one of the top stories of 2012. And it really helped put this topic back on the map and made it part of UN high level meetings. So we did a lot of science. There was a whole series of papers, but I want to break the science down Fisher-Price style for you. So what we learned, using genomics to study the evolutionary history, is that ST398 actually started off as MSSA, methicillin susceptible Staph, so not resistant, so no lightning bolt, in people. So it was an organism that was spreading in people. It jumped from humans to pigs, probably 30 years ago. We're still trying to get the molecular clock calibrated for that. And it was in the pigs, it was in the food animals where you have rampant antimicrobial use that you see the transition from MSSA to MRSA, so going to resistant strains. And we also see it's not just methicillin resistance, but tetracycline resistance was almost universal, which is the most commonly used drug in food animal production. So we get the lightning bolt, and then it starts, see this very faint arrow from humans to pigs, bold arrow from pigs to people working in the industry, bold arrow from them to their household contacts. And then we also started seeing it into community contacts as well. So is this secondary exposure to animal production? Is it from the application of animal waste? We still don't know. Or is it, secondary human-to-human transmission? But then because we had the genomic markers for the livestock-associated strain, we could trace the strain spreading into hospitals, and we saw outbreaks in hospitals of the livestock-associated strain. And we also saw lots of food contamination and evidence of foodborne transmission. Not a lot of foodborne transmission, we don't think. So food does not seem to be a really good vehicle for Staph aureus, which tends to colonize the nose and throat. But we did see clear evidence of this happening. So this was a real paradigm shift in terms of what MRSA could look like. So originally, MRSA was a hospital or a healthcare associated thing, right? You got MRSA in the hospital. And then later we learned through molecular analysis and lots of epidemiology that people learned here at this school, we learned about community associated MRSA. So think about locker rooms. So the kids coming home with these drug resistant skin infections. But now we have LA MRSA, livestock associated MRSA. And this is now a textbook case of what can go wrong when you use antibiotics in food animal production and you raise animals in this crowded, unsanitary conditions, because we know that that's how staph spreads. So that was staph, but the organism that I've become obsessed with, and my daughter loves this stuffy too, is E. coli. So E. coli ranks among the deadliest bacteria in the world. It kills 1 million people around the world. And it's the #1 pathogen, the top pathogen associated with fatal antimicrobial resistant infections. And when the general public hears about E. coli, I think they think brown, right? So they tend to think about diarrheal infections. But that's only 6% of the global deaths are associated with diarrhea. They should be thinking yellow and red. So it's urine and blood infections that are killing most people. So 94% are extraintestinal infections. So especially UTIs and bloodstream infections. And it turns out it's the people living in low and middle income countries that suffer the worst from these. So 80% of the deaths are happening in low and middle income countries. So this is a social justice issue, like in your face social justice issue. This is a big deal. But despite E. coli's dominance as an antimicrobial resistant pathogen, we really know little about its ecology. We don't know how these guys get around. Where do they live most of the time? And how do they get to us? And I think understanding this is key to decreasing that, let's bring that number down from 1 million people, right? So one thing we do know is E. coli is super versatile. It can live in, most terrestrial vertebrates. It can live in their guts, including all the major food animal species. It can also live in our super cool clip art guy here. I love the new clip art people. So we know that vertebrates, including humans, carry E. coli. Most of you are carrying it today. And we also know then that while there are thousands and thousands of strains of E. coli, from a clinical standpoint, there's really 3 types. There's the good or commensal kind that you're all sitting on now. There's the bad kind. So think about the Jack-in-a-Box incident of the outbreaks, in the early 90s and the frequent lettuce, romaine lettuce outbreaks. So those are the dirogenic E. coli. They kill in the United States, less than 100 people today because of a lot of interventions that we've had. But the XPEC, or extraintestinal pathogenic E. coli, that cause UTIs are the ones that kill the most people. 36,000 to 40,000 people a year in the United States alone. So our good friend and colleague, Jim Johnson, coined this term, extraintestinal pathogenic E. coli. For the kind of E. coli, they can live in your guts without any symptoms. So it's not a pathogen in your gut. But if it gets into the urinary tract, it can kill you. So it has these special virulence factors that can cloak itself from the immune system. It can attach to your erythroid cells, even under the sheer force of urine. They actually hold tighter. They can acquire iron and deal with uric acid. They have all these unique features that then allow them to cause 6 to 8 million bladder infections in the US each year, over 250,000 kidney infections, and as I said before, 36,000 to 40,000 deaths due to bloodstream infections. And antibiotics are critical to prevent this ascent from the bladder to the kidney to the blood. And if you fail at any of those steps, you could lose the patient. I just have to underscore the urinary tract is the primary gateway to the blood. So we can't dismiss urinary tract infections as just, benign annoyances. And I want to lock this relationship in for you. Most urinary tract infections are caused by E. coli, 85%. And it's usually the E. coli in our guts. So they live in our guts without symptoms. And it's when these E. coli get from our own anus to our urethra that we get that infection. Sorry to be graphic, but sort of my style. And we need to understand this, right? So most expectant infections are sporadic. So they're not these dramatic foodborne outbreaks, but it's just when that trip happens, people get the infections. But it's a sporadic infection that's happening a lot. As I said, 6 to 8 million a year in the United States alone, hundreds of millions a year around the world. And so we've been asking this question, how do these get in our guts? And so we know that who we live with can really increase our risk. So human-to-human transmission is important. It's probably the dominant way that we get it, either directly or because the world's covered in a thin layer of poo, we're sharing microbes all the time. Are you still supporting this lecture series? I hope so. Sorry. Maybe I wasn't the best person to start this new series off. Sorry. So we've been asking this question though, what about this route? Can people pick up these E. coli from food animals? And either from direct exposure, through occupational exposure, through contaminated environmental media like animal waste into surface waters, or through meat as a vehicle. And so, we started studying this, and this was Cindy and I started this work. And it's embarrassing to say 12 years ago we started, because it took so long. This was my albatross for years. So we started in Flagstaff, Arizona. So we were living in Flagstaff, working at the Translational Genomics Research Institute. I was missing Baltimore a lot, and I was thinking about all the great research that I could do in a big city. And then I realized I was living in almost the ideal place to start this research, because this is a small, geographically isolated city, 70,000 people, and there's one hospital. And my good friend, Lori, was the head of infection control there. And so she said, let's do a study. And so we spent a year collecting all of the urinary tract infection isolates and all the bloodstream isolates from the only hospital in the city. And then we went to every major grocery store chain in the city twice per month and bought every brand of chicken, turkey, and pork. Brought it back to the lab and cultured it. And at the end of the year, we collected and sequenced nearly 2,000 from meat and nearly 1,200 from people. Now, I was feeling pretty confident at this point. We'd used genomics to identify the origin of the Nepalese, of the Haitian cholera epidemic. And we'd done the work on Staph aureus, and I was like, yeah, no problem. And I had this plan. We were just going to figure out what is that Venn diagram? What's the overlap? And so initially it was this 3-step process to sequence the genomes, to group them by this standard genotype, the multi-locus sequence typing, so sequence ST, sequence type. And then we would do these core genome phylogenies, or evolutionary trees, that we'd been helping to pioneer. And I was, like I said, feeling pretty confident. But then we started getting the sequence types back. There were over 440 different sequence types just in our collection. And so each of those numbers is a different sequence type. And you see the size of the bubble represents how many isolates are, each time we collected an E. coli, how many had that different sequence type. And what you see, if they're red, that's scaled for the number of isolates that were from meat. If it's yellow, it's from people. And so I was pretty excited initially because, we had a lot of overlap. So while you see that some strains dominate in food animals, so, ST117, for instance, others like ST131 dominate in humans. But we had these mixed pie pieces. And so we decided to start with the one we knew best. We'd done a lot of work on ST131. It's a pandemic strain, kills a lot of people around the world. And most people had said there was no involvement of food, but we see this big pie piece. So we took that one, we took the genomes and we started drawing these family trees. So this is called an unrooted tree. And what you see is this big cluster up at the top, let me circle that for you, is the pandemic lineage. Well, we kind of held up what they said. There's no food involvement. You don't see any red squares. Those are all human infections. But we saw this other cluster down here, the H22 lineage. And this lineage is also a big killer, right? So it's an important pathogen causing infections. See all the yellow dots. And so we wanted to understand how they were related to one another. And so we just isolated those, drew another tree. And what we saw were these clusters of really closely related meat and humans. We thought we nailed it, right? So we sent this off for review and the reviewers came back and said, how do we know it wasn't the people giving it to the chickens, right? How do we know people weren't contaminating the meat? And in fact, we didn't have any clonal relationships. So if they were, sorry, if the yellow and the red boxes were sitting on a flat line together, then we'd have like perfect clonal relationships. So that'd be pretty good evidence. But what we saw was there's lots of distance in these trees. And so I realized we were just struggling with all of the enormous diversity of these E. coli animal populations. And I shouldn't have been surprised because, you know, we're raising chickens like this. Every one of these chickens has E. coli. And that's 100,000 birds in a single house. If you had to define the E. coli in that population, how many samples would you want? I'd want a lot, right? This is the map of the United States. Each of those blue dots, those tiny blue dots, is 1 million chickens. That's annual production in the United States. So we're producing billions of animals on thousands of farms. Now, how many samples do you want? I mean, it is impossible, literally impossible to sample enough to define the E. coli populations. And what I realized is that we are using these tools best suited for outbreaks. So an outbreak is like this, right? You have a new pathogen that's emerged on a farm or in a slaughter facility and in one place, and then they start contaminating meat products with that pathogen, and then they start disseminating those across the country. Well, those can be traced back then based on epidemic curves that we all learn about in School of Public Health, right? And we can trace them back on, you know, using shoe leather epi. But this is not what we're seeing here. What we're seeing is just continuous spillover of E. coli from all of these animals that are colonized with. These are old, diverse E. coli that are just spilling over all the time. And if I drew all the red arrows, that would be a very red map that would make any Democrat very nervous this time of year. So, but what I realized is in the case of XPEC infection, our challenge was not to pinpoint a specific farm or slaughterhouse. It was to determine whether it was food animal or human, right? So we needed to go a step up a level. And so most of the epidemiologic investigations are performed using what we call the core genome. So all of these E. coli share a chromosome, and there's portions that they all share, and we look for little mutations, and we can use these mutations to draw these evolutionary trees. But it's the core, it's the accessory genome. It's the pieces of DNA that come in and out that lead to niche adaptation. So think about, adaptation to a person, or adaptation to chickens, pigs, turkeys, etc. We think that it's these flexible genes that they're pulling in to adapt to those different hosts. And so that's pretty cool biologically, but I think it's really cool if you think about it as an epidemiologic tool. So if you have, an E. coli emanating from a chicken host spills over into a human host, well, we think initially it's going to look exactly like that. It's got all these chicken, plasmids or mobile genetic elements. But over time, it's gonna adapt to the new host and it's gonna look different. The chromosome might look the same, but these extra pieces of DNA are gonna look different. So what we can do with this information is we can differentiate historic transmission events where we can't do anything about it anymore, right? It's established now in humans versus those that just happened, right? So those are the strains that we want to go after. Those are the spillover events that are happening today. And so what this made me think of is Forrest Gump. So again, I'm a pretty simple dude. I remember sitting on the park bench, and he says, "Those look like comfortable shoes." Mama said you could tell a lot about a person by their shoes, where they've been and where they're going. So what does she do for a living? She's a nurse. I mean, we know that, right? And so I'm looking for the markers, the shoes that chicken wear, that E. coli wears when they're in chickens, right? And so we started looking at the accessory genome of E. coli, and working with our friend Tim Johnson, who was an expert in plasmids. He said, have you looked for the Colby plasmids? And so we looked, and turns out that 63% of chicken and 64% of turkey carried these plasmids, these mobile genetic elements, these pieces of DNA that bacteria can bring in and out, and they can exchange. Can exchange. Only 5% of human E. coli had those. So I think these are the shoes that E. coli wears when they're working in chickens, right? And so what's cool about this from an epidemiologic perspective is that you get somebody with a urinary tract infection, you collect the urine culture, and then you find the E. coli, and you find these chicken shoes, and you can say, hey, this came from chicken, not another person. So we went back to our challenging tree here, and we overlaid it. We put these chicken shoes onto that tree. Do you see what happened there? So all of the human infections that were most closely related to the poultry isolates had these plasmids. And so even our harshest critics had to admit we nailed this. And so this has been another highly cited paper. But I knew if you look at the massive genome of E. coli, there can't be the only one. And so we found 17 more of these host-associated mobile genetic elements, pieces of DNA that they could bring in. So more shoes. And we found 11 that were associated with one or more food animal species, and 6 associated with humans. And then my great colleague, Dan Park, and our colleague from University of Michigan, both former Hopkins people, helped develop this Bayesian latent class model. So we could use these host elements along with other fingerprints in the DNA to predict the probability that an E. coli came from humans or animals. And so let me tell you, this is all, again, Fisher-Price style, because I think pretty simply about this. And Keith always makes fun of the way I say Bayesian. Anyway, so here we go. So a person, infection, we get the E. coli. Well, we can start looking at information. So we want to know whether it's human or food animal. There's an initial probability that it came from food animals. And so we can just look at that. But with these Bayesian models, we can start to incorporate additional information. So we can do a sequence type. Maybe a sequence type 69 shifts us a posterior probability towards food animals. And then we can start going through our different elements. And those are not the icons that I had, but it's fine. So we can see that but as we go through and look at these different shoes, we can determine that this strain almost certainly came from food animals. And so we actually have a quantitative number now. And so we cut it off at 80%. So if it's 80% better probability of coming from food animals, then we say it came from food animals. And 80% better probability from coming from people, we say that. And so we started looking at the thousands of E. coli that we collected from people and from meat. And what we saw was that about 8% of the E. coli, the urinary tract infections, had 80% better probability of being from food animal production. Now that might sound like a small number, but that translates to over 500,000 infections a year across the United States. We also applied it to the food isolates, and 0.5% of those appeared to be coming from humans. So it's not from humans to animals, it's the other way around, right? And so we call these foodborne zoonotic E. coli. And so now we have this number. And we published these data, and it really got a lot of media attention, this number of 500,000 UTIs. But Flagstaff is a young and homogeneous population. So it's pretty socioeconomically and ethnically pretty well off and white. So what about in more diverse states? And so we've been doing a studying something called SB27 in California. And through that study, we collected and sequenced a lot of genomes from people and from meat. And this is work with other former CLF fellows. That's your photo from when you were a kid there. Anybody recognize Keith Nachman there? All right, Joan Casey. So this is what's so cool, that we're all collaborating today. So fast forward 17 years later, and we're submitting grants and doing collaborations together. And so we've started running the model on these. It's 18% in California. And so about one in five of UTIs in California appear to be from food animals. And so we have this question, like, why are these so much higher, more than twice the rate of Flagstaff? And so our colleague Dan Park has been studying this, and it turns out that there's, the FSEC rate, the rate of zoonotic infections is clustered in specific spots, and it's also clustered in the same spots as poverty. So there's this dose-response relationship. So with every 5% increase in poverty rate, there was a 14% increase in zoonotic transmission. So that's in America, where we have good water sanitation and hygiene. I think that these could vary greatly depending on cultural practices, water sanitation, hygiene, or WASH, animal production, like how intensive is animal production? And so what about low and middle income countries? Now, this is from a paper that we published with Maya Nanapali, where we were looking at E. coli transmission and drug resistance in Cambodia. In Cambodia, you have this just really rampant uncontrolled antibiotic use in human medicine, and you have almost industrial scale or intensive farming. You have extensive farming, again, antibiotic use. You have human waste being flushed out untreated into surface waters that are being used to grow food. You have open defecation. You have fish farming. You have animal waste being flushed out untreated. So the ability for bidirectional sharing of drug-resistant pathogens couldn't be more sort of in your face than in Cambodia. And so again, this is Maya's work. And what she showed, well, let me contrast it first. What people from high-income countries have been saying, like the UK, this was a very influential study. They said they looked at these ESBL genes, these drug resistance genes, and they looked at the potential sharing between meat or food animals and people. And what they showed was there was really minimal sharing in the UK. And this was a really influential study that caused people to say, yeah, maybe it's not such a big deal. But then let's look at what's happening in Cambodia. I mean, it appears that these genes are really flowing between food animals and people. And so it's just contextually, it's very different. And what I think we're dealing with is a high income country bias. Most of the money and research is happening in high income countries. And so we don't have the data. And we need to get the data to really, if we're going to form policies and put this in a global perspective, we need these data. So we've asked this question, are zoonotic expat infections more common in low and middle income countries than high income countries? So we've been applying this model. Well, we don't have the samples, but we really want to look at bloodstream infections, right? Because that's what's killing people. So we went to these public databases, and it turns out there's thousands of E. coli genomes in these public databases. And so I have this great team. Cindy and I have a great team that she's really built at GW, and so they've been Maliha and Yashan and Edward and Dan have been scraping public databases, and they've got over 3,000 bloodstream E. coli genomes and over 8,000 from meat. And we started applying our model to those collections. So let me show you what we found. We found we have strains or genomes from high-income countries and low-income countries. Yay, right? This is great. Look at this. This is a travesty. 93% of the genomes are coming from high-income countries. These are the people living in low and middle income countries that are at greatest risk for dying of these infections. So 3 times the risk, and yet we're not sampling there. We have to address this. But let me show you what we found when we ran our model. So we found that about 17%, this is holding up with our California data, 17% of people in high-income countries are their UTIs and bloodstream infections are appear to be zoonotic. 29% low and middle income countries. So 1 in 3 bloodstream infections might be from food animals in low and middle income countries. This is really important to know. And these strains were extensively resistant, 3 times the risk, 3 times the rate of resistance to 10 or more classes of antibiotics. Classes of antibiotics, right? These are really resistant. Now, we have to look at the data with, we have to look at the data carefully because they're undersampled. We don't know the bias going into those sample collections, but it is underscoring we have to look more. Because if you translate, if you adjust for the proportions that we saw there to the world's population, and then you look at those million people that are dying, we estimate that it could be 250,000 people dying of zoonotic E. coli, so E. coli from food animals, that are currently unattributed to those sources. This is a big deal, and this would change the way we would try to intervene, right? It gives us new avenues to intervene. So we could potentially identify the highest risk strains. We know only a few, it's a small number of strains that are actually making their way over. And so we could develop vaccines for animals. So vaccinate the animals against the highest risk strains to try to reduce that transmission. That's just one possible way. Probiotics, phage, these viruses that can infect and kill E. coli. And so this is one of the things that I'm going to spend the next 10 years of my career working on. And I just want to highlight something, which is that the world has caught up, right? So the FAO, the OIE, the World Health Organization, the UN, they talk about something called One Health. This is this recognition that human health, animal health, and the environment, health of the environment is inextricably linked. This is what Bob Lawrence has been telling us for 25 years, right? So they're catching up to you. They're catching up. And this is what you taught me. And so I feel like being at a Center for a Livable Future really set me up to become, a leader in this realm. And I'm really proud of the work we've been able to do. So because of the kind of talk this is, that's my science stuff. So now I get to pontificate a little bit. All right. So, because I have the microphone. So I think it's, one of the things that I learned here was that, I wanted to work on policy. I really wanted to work on health policy. And my advisor said, don't study policy. You stay on track, keep doing microbiology. They'll invite you to contribute to policy if you stay in the sciences. And so I did that. And what I've spent the next 20 years doing is really balancing these three things, right? So research, science communication, and policy. And what I realized is that when I started doing these Hill visits and the industry had visited the congressperson before I got there and told them all the things I didn't know, they would tell me those things. And then we go back to the lab and try to close those knowledge gaps. And so you've just got to sort of work iteratively through this. And you never, policy is really fickle, right? You never know when something disastrous is going to happen and you can't get anything passed. And I think that this is the way policy looks most of the time. This is Death Valley, right? Policy looks like this all the time. It's just you try and you try and you try. And if you're just doing that, then you're gonna burn out. But if you can go back to the lab and keep working on it, then it's gonna rain someday and you have these massive blooms, right? So you have these periods in Death Valley where these flowers go. And so I feel like this is what the CLF fellows are doing and have been doing for 20 years. And this is what I think I've been trying to do is continue to plant those wildflower seeds and wait for the rains, because you can't make the rains come. And planting seeds is things like, original research, agency meetings, Hill visits, go meet with the industry leaders. We've helped turn the industry. I mean, poultry production, from the time I graduated till today, we went from a tiny fraction produced without antibiotics to more majority produced without antibiotics, at least antibiotics important for human medicine. You do press releases on your science. Science doesn't speak for itself. You have to translate it to the general public. Science communication, Fred Kirschman, he showed me, you've got to be clear. And whenever they invite you, you go do hearings and briefings and you just keep talking about it. And then you do documentary films, and CLF is doing all of this stuff, and they taught me to do all this stuff, and it's been an amazing journey. But I think that this is where I'm rooted, though, is that continuing to plant seeds with the original research. Okay, so now my life. Now just a little bit about life for me. So I kind of think of life as a little bit like a pachinko game. People know pachinko, like these mechanical, machine. You shoot up a ball and it kind of hits these things. Apparently, there's a series. Sean was telling me about this. I think my life was, so you don't want to go down there. So you don't want your ball to go straight down there. You want it to go into one of these other little catches and you win lots of balls. But I think I was kind of set up for this journey, right? So when I was 11, my sister got married. She got married at 18, left the house. My father died. My mom slipped into a depression that she didn't ever recover from. And I kind of went feral. So, I mean, I went feral, and I had always struggled in school. I got expelled from high school, had to do an extra year, and I went to welding school. And I learned a vocation, and I worked for a couple of years as a welder. In Phoenix, wearing leather outfits, welding is a really crummy job. But I had people who believed in me, and I had a boss that said, you need to. He was like one of these pins. He said, Lance, you don't need to weld. You need to go to school. But I was too insecure. I'd been a terrible student, and I was too insecure to fill out the form. And Lois Stansbury, my friend's mother, helped me fill out the form, and I got in. I don't know how. I'd gone to community college a little bit, right? So I got into college. Then I had this guy, Dick Shand. I was taking microbiology, and he was so into micro. He is such a nerd, and I got bitten by it, and I asked him if I could work in the lab, and he let me work in his lab. And we fought a lot, but I worked in his lab, and I learned so much. He was such a micromanager, and I learned so much. And then this guy, he really believed in me, and he took me after my master's, and I started working in his lab, and I really started learning about genomics, and he's been a friend and mentor to me for 30 years. And then I met this lady, and I got on an elevator with her. I was working. I'd moved to Maryland. Let me just tell you this: I moved to Maryland to do something about antibiotic use in food animal production. I was a microbiologist, and I wanted to have a positive impact on the world. And I had this. I just was like, I need to move. I moved with a friend to Maryland. I'm like, I'm going to work on this, and I was working in a lab at University of Maryland, and I got on the elevator with this lady who was just talking as loud as as loud as she wanted, as if everybody wanted to hear what she had to say. And she started talking about antibiotic use in food animal production. And that's exactly what I wanted to work on. And it turned out I knew a lot about anthrax. And I developed genotyping system for anthrax and plague. She wanted to work on that because that was the latest kind of shiny thing. And I said, I'll come help you with that if you help me do this. And she brought me to Johns Hopkins. I would have never gotten in through the front door, I can tell you that. She brought me in through the back door. Pat Bricey, when I submitted my GREs, he's like, you should have kept those to yourself. But as usual, Ellen didn't really have a plan. She had no real plan. And these two were more pins than that pachinko machine, right? And these two, Carol and Rhonda, made sure I didn't fall through. And so I started working with her. And then she said, you need a PhD. And she crashed me into the program. And then I met you all. And these were three big pins in my life. And you really helped push me into one of the big slots where all the balls come out, And it's been such an amazing journey. And your belief in me and your support was so critical. And so these CLF fellowships, I mean, it's just amazing to me. If you think about the ripples that you're sending out, the sparks that you're sending out into the world for change, for positive change, people who are basing themselves, they're, that are basing themselves on sound science to go out and make real change, it's amazing. And I can tell you, like, 20 years later, this was my crew right here, right? Sharon Napier, she's at the EPA. She's a lead scientist there. She's been the first person to really start looking at antimicrobial resistance in water and been trying to push the EPA into the National Antimicrobial Resistance Monitoring System. She's carrying the torch. Jay Graham, this guy's amazing. He was at GW for a while. He got me recruited to GW. Then he went to Berkeley. Now he's a professor at Berkeley. He's running the MPH program, online MPH program. But really importantly, he's doing all this work. He puts his life on the line, going to Ecuador and other places to study how people are getting infections, people living in peri-urban communities. He's always put himself at risk. And he and I have a grant together. So we're still working together. And I have a grant together to study UTIs in Ecuador. And so we're starting to close that knowledge gap. And Keith I don't know what Keith's doing these days. Keith, Keith was just my buddy when we were here. He was just my classmate. But we got a grant together working with Sarah Tartoff at Kaiser Permanente and Joan Casey. And I've been blown away by you. I mean, you're amazing. He works so hard. He's such a joker, right? But he works so hard and he's so smart and he's so diligent. And he doesn't let stuff fall through. All right, maybe some paper edits, but he's amazing. And he does it all the time with sending inappropriate texts. And it's just awesome. And so this is part of the legacy of this center. And it's so amazing. And I don't know if I can do this. This is the other person that I met when I was here. She took a year off of medical school to get her MPH. She's the smartest person I know, the hardest working person I know. And I would not be here today. I would not be at this podium if it wasn't for her. I mean, she is an ace. And she's both so supportive to somebody who can be a real jerk at home, but also just like her ability to I can say I want to go over there. I can have vision and I can turn on the Riz sometimes, but she knows how to play chess, right? She knows how to put the pieces in place to get us there, right? And I couldn't get there without her. And she deserves this honor as much as I do. So Cindy Lou. So I'm really excited for the newest cohorts of CLF fellows. I don't know if I can promise you love and marriage, but I think it's a high likelihood that you're going to have friendships and synergy for the rest of your lives. And I love my colleagues that I met here and I continue to work with. And I just have one other peg in my board. There's two of them back there. There's Sarah, who's a peg, and then there's Gabriel Goodenough. So this guy is amazing Baltimore booster. This guy is dedicated to this city, and he made this amazing, beautiful film called The Body Politic about gun violence and our mayor trying a public health approach to stopping gun violence in this city. And no more, not the short term, let's put them in prison, let's give them rough rides. Let's get out in the street and change things. And Gabriel documented that work. And Gabe, I love you, and I really appreciate the work that you've done. And so you all have to see this film. If you care about Baltimore, you have to see this film. And so with that, I just want to thank you all and thank you for this honor.
Lance, thank you. I think I can speak for everyone. That was a wonderful presentation, and we all appreciate your sharing your personal story as well. We have plenty of time, 10, 15 minutes for Q&A, and Dr. Nachman is going to facilitate that Q&A. And we have mic runners, I believe, Allie on the left and Deanne on the right.
I was going to Phil Donahue this, but I guess if Dee is here, I can pass it to her. All right. So any questions for Dr. Price?
That was fantastic. Thank you. One question for you. I totally buy that it's coming from animals to humans, but through the means, through contamination, what route?
Yeah,
Do you think is the major contribution?
I think it changes with context, right? So I think that in the US, it's almost all coming from meat. You know, it's really hard to handle raw chicken in your kitchen without spreading those bacteria, right? I don't know. This is, you open that package, you're almost certainly gonna. I've tested a lot of chicken, right? There's bacteria on there, lots of drug resistant bacteria. There's no doubt that it gets around. Cindy and I are watching each other like hawks whenever we handle raw meat, and I guarantee there's some of those bacteria in our kitchen, right? And so they get around. But in low and middle income countries, I think it's probably water, sanitation, hygiene. That's the poor infrastructure there. And also intensive human animal interface, right? So some of the communities where Jay is doing his work in Ecuador, people have the chickens living under the beds, right? And then, I think so. And as I showed you those pictures from Cambodia, I think it's just, we're just sharing all the time. And so when we're talking about, a lot of people, when they're talking about, intervening in the antimicrobial resistance crisis around the world, they keep talking about, antimicrobial resistance or antimicrobial use goals, reductions. But we have to be thinking about water and sanitation and hygiene infrastructure. And that's really World Bank scale stuff, right?
So one other question, if I might.
You're the chair.
You did all this genomics, and so my question is, what are there particular gene families that tend to be a target for this?
Yeah, so
Understand what I mean?
yeah. So it's interesting. They're different dynamics, right? So what we showed, we did some other genomics on that ST131 clone. I told you about that pandemic lineage, right? A lot of people thought that was actually an epidemic of these CTXM genes, that they were spreading among E. coli, and they are mobile, but it turns out it was one, it's called a clonal expansion. It acquired it once, and then that strain just caught fire and took off around the world. But there are other dynamics where you see, it seems that there are genes that just really do hop, right? So there's the MCR1 gene that was clearly emerged, or at least it was really amplified in food animal production in China. And that one seems really promiscuous. Like we kept finding in all kinds of different strains. And so I think it's a different dynamic. And so that takes different tools. I don't know how we really go after when it's the gene that's the epidemic. That's scary. We know how to control individual strains. We have to figure it out, and then we have to go for it.
Well, it might open up some therapeutic options.
Yeah.
Very nice, thank you. You showed pictures of pigs and cows and chickens, and follow up on her question. It's not for me because I've been a vegetarian 50 years, but should people be more worried about their pork or their chicken or their beef in terms of transmission?
It depends on the bug, but we're still trying to. What's that? For these E. coli, we're still doing the quantitative analysis, because you have to scale for exposure, like how much meat people are bringing home. We tend to see more of these in poultry, though, but I want to hold my answer until we've done the formal analysis.
5 billion chickens per year in the country or something.
9 billion.
9 billion.
9 billion chickens, 300 million turkeys-ish. 100 million hogs, 50 million, 30 million cattle. I don't know. You guys would know the numbers better than me, but that's But on a pound per pound basis, those actually translate, except for turkey, they translate to, roughly the same amount.
More questions? Tom?
Is there a mic?
We have room mics, actually.
We have ceiling microphones, so if you just speak loudly. Okay. Yeah, so great presentation. Thank you so much. And I remember the paper about E. coli association with UTIs. It came out a decade or so ago. In the past decade, we have seen a reduction in poultry in antibiotic use. And I wonder if there's been updated studies and if UTIs associated with poultry have gone down, or has that research been done?
Well, so actually what we see is more, we studied two different sites is the problem. So there's Flagstaff, where it was 8%, California was 18, right? But I think what you're getting at is, what about drug resistance rates? Maybe. Yeah. We haven't done we haven't done a direct formal comparison between the two, but overall drug resistance rates have been going down in poultry. I guess we are doing that formally because the original purpose of that study was to study at Senate Bill 27 in California to see what was happening if you decrease antibiotic use in food animal production, or at least you have a policy that's supposed to do that. Will you see reduction in resistance? What we tend to see is that across the country, antimicrobial resistance is decreasing in poultry, which kind of makes sense because that's where you've seen the most progress in terms of reducing antibiotic use and retailers voluntarily saying, we're going to stop doing this.
We have a question online, Lance, that I read to you. Large-scale industrial CAFOs are proliferating in lower and middle-income countries, and animal production on smaller scales is often getting more intensified as well. Would we expect antimicrobial resistance to behave differently between these two environments?
Good question. So I think, you have to look at the environmental context, like what's the water source? Interesting. It really depends on antimicrobial use being a primary driver, but then what kind of microbes are the animals getting exposed to as well? So one of the things that Jay has been working on is looking at irrigation, contaminated irrigation, carrying drug-resistant bacteria into communities that don't use a lot of drugs in their animals, but the animals are picking up drug-resistant bacteria from that contaminated irrigation. So I don't have a direct answer for that.
Great. I think we have time for one more. Dr. Schwab.
Dr. Schwab.
Dr. Price. Excellent talk.
Thank you.
Forecasting is dangerous, right? But it's kind of cool. You do a description over 20 years. You've got it. You're starting to go into sequencing. What do you see happening in the next 15 to 20 years with both technology for enhancing your approach, citizen science out into these things with sensors? And then how is this going to translate into policy in other countries too, do you see?
Yeah. Well, what I can tell you is our biggest hindrance right now is the cost of sequencing. It was going down really fast, right? So with these massive parallel sequencers, it was going down really fast, and then it just hit this plateau, and it still is costing us about $100 per, between $50 and $100 per isolate. And so if we can drive that down, and I'm sure it will be driven down at some point, I hope, then we can start to collect the samples and sequence them and really get those fine level answers that we need. So that's one of the things that I'm looking for. I think the other thing that we saw with COVID actually was, these new vaccine technologies. And so what I'm really excited about in the next phase of my career is using the genomics to identify the highest risk zoonotic strains, but also the highest risk human strains, and then using these tools to develop new vaccines that can be applied in animals, be applied in people to try to drive down transmission. Probiotics, I think actually the magic combination is going to be sort of try to slow them down with both vaccines, maybe some probiotics and some phage, hit them in 3 different ways. But we need cheap, easy to deploy technologies for low and middle income countries. And so I'm hoping that that those will come as well. I guess I just told you what I'm hoping for, not really what I see, but it's as good as anything. I'm about to go on sabbatical. Cindy and I are going on sabbatical in Denmark, and so we're going to spend a year at the Danish CDC, Staten Serum Institute, and at the Danish CDC is also the International Center for Antibiotic Resistance Solutions. And so I'm hoping to do a lot of work with them, and what they're doing is exclusively working low and middle income countries. And so I'm really excited to push them on trying to develop new ways to go after E. coli specifically. I think we can save a lot of lives.
Turns out that Dr. Price is so efficient at answering questions, we have time for one more.
This is not usually I'm usually really long in my answers. Yes.
The question,
Yeah, at the end.
go ahead.
Thank you so much. I wanted to harp back on one of the points you were making about the disparity and where the research exists and doesn't exist. And my question to you, having seen that kind of disparity and where the state of the world is right now, what can we do in higher income countries, institution higher learning in the United States and other higher earning nations to help mitigate that disparity, to increase the research, to increase the data in lower and middle income countries so that we don't have this kind of
Yeah, we have to find partners. So we found partners in Nigeria and Ecuador. We're going to start reaching out in other places. We start writing grants with them to get funding to support them, and don't keep the money here. Send a lot of the money there so that they can collect the samples. Do exchange programs where we bring them over to train them on the work, teach them what we're doing, and so that they can bring it home and do more work in their own institutions. And so it's capacity building, but also just real genuine partnerships. And so that's what we're trying. And we're working, again, one of the things that we're trying to do is work through ICARS. ICARS they're in 20 different countries or more, and only low and middle income countries. And we're going to say, let's work together to get more of these samples and start closing this knowledge gap. So yeah, just outreach and get out of your comfort zone and go out there. I'm afraid of foodborne illness myself, but I'm gonna go. I'm gonna do this. Thank you for that.
Okay. One more time, I want to thank Lance for one. I do want to thank everyone for their questions and also just say one thing to Lance about your feralness. I don't think you've lost all of that. I'd actually say you're among the finest feral public health advocate researchers I know. So just want to make sure that's on the record.
Thank you.